So, structure number 1. What's the purple stuff with the little dots on it? Please. Rough ER. Okay, and the job of the rough ER is to transport proteins. 1 mark for the name, 1 mark for the function. No. Okay, number 2, we're looking at the purple stuff with no dots on it, which would be smooth ER. And I will accept for smooth ER, produces lipids, repairs membranes, makes hormones, detoxifies poisons, and breaks down complex carbohydrates. Any one of those would be acceptable as a function of the smooth ER. All right, number 3, pointing at the nucleus. If they wrote chromatin, that's fine as well, as that's what's in there, if they were confused on that, but always go with the labels I gave you on the cell diagrams the day we labeled them. Okay, so we're looking at nucleus here, okay, and the nucleus contains the DNA. Okay, holds DNA, something like that. Okay, that's the reason why if you say chromatin, then you really don't have a function, because chromatin is what DNA is. They're the same thing. Okay. All right, number 4, the darker dot in the middle. Nucleolus. Spelling counts on that one. Okay, and the job of the nucleolus is to control protein synthesis, copy DNA, okay, transcribe DNA, something like that. But technically, it also does that, yes. Yeah. Yes. Yeah, produces RNA as well. Yeah. All right, number 5. What's the outside of the nucleus called? Nuclear membrane. They may have written nuclear envelope as well. I don't really like that as much, but you can go with it. Okay, and the function of the nuclear membrane is essentially to allow RNA out, okay, or allow DNA to, or copied DNA to get out, okay. Control movement from the nucleus, although not much goes into the nucleus. All right, I don't really like that, but we'll go with it. Yes, that'll be fine. Yep. I don't like protects for membranes. I don't like that. No, I'm going to say no on that. No. Protects, no. Have me look at it after. Not a wall, no. Okay, that'll accept. Yeah, okay. All right, for number 6, the little dots are ribosomes. Okay, and what do ribosomes do? Please. They make protein, produce protein, synthesize protein, something about protein and the making thereof. Okay. No. No. Okay, number 7. What are we looking at there? That is the Golgi apparatus. All right, and the job of the Golgi apparatus is to package wastes for removal from the cell, treat wastes, make vesicles to take waste out of the cell. We also said it produces peroxisomes and lysosomes. Those would also be acceptable. Okay. All right, number 8. Cell membrane. What 2 words do I need in the description of what it does? Right, endocytosis and exocytosis. If they have something about those 2 things in their answer, it's right. Number 9. Yeah. That's the mitochondria. And what does the mitochondria do? Carries out cellular respiration, burns sugar for energy. Okay, something about energy. Yeah. Yep. Okay, and number 10, okay, is pointing at these things here, which would be lysosomes. I will also accept peroxisomes, although there is quite clearly stuff inside of both of these. Okay, so we're going to go with lysosome on that. Okay, but I'll accept peroxisome if they said it. And what's the job of the lysosome? Yeah, Leah. Digest food, other cells, digest stuff, break stuff down. Yep, also does that. Okay, there's something about digestion, that's good enough. Okay, and number 12, stringy things. Whoops. Well, that's because it's up here. I'll get to it. Number 12. I'm going in clockwise. I'm not going in numerical order. Microfibers, microfilaments, microtubules, cytoskeleton. Okay, I would say I'm going to write cytoskeleton because it covers all of those things. Okay, but if they have any of those, okay, that is fine. Function of the cytoskeleton is to support the shape of the cell, allow the cell to be able to change shape. Something about cell shape. That's the centrioles. That's number 11. We're doing number 12. Cytoskeleton doesn't. Don't look it up on the internet. Listen to my podcast. That's the only internet thing you should look up. Well, no, that's kind of implied in the name. Not good enough for a function. Okay, and then number 11 is the centrioles. No. Centrioles and the centrioles help with cell division. Okay, that's their job. So if they have something about cell division on there for centrioles, that will be fine. Mark out of 24, please. Give it back to them. Make sure your name is on it. And put it in the box. Okay, so we're talking about prokaryotes and eukaryotes, which are essentially the different kinds of cells, okay, that living organisms can have, right? Eukaryotes are organisms like ourselves. They're cells like the one we just finished labeling, okay? They're cells that have organelles inside. They have compartmentalization. We talked about that before, okay? And then there are the prokaryotes. Prokaryotes are much smaller. There are no multicellular prokaryotes, okay? And their cellular structure is, as a result, quite a bit different. Okay, first off, there's no real true organelles within a prokaryotic cell, so there's no compartmentalization. Okay, they have cell walls, but the cell walls are a lot different than plant cell walls. They're made of different materials. In fact, that's how antibiotics generally kill them, is they make their cell walls break down so that your immune system can destroy the cells more easily. Okay, that's what penicillin does. We'll talk about that in a little bit. Okay, so we're going to be going over the differences between prokaryotic cells and eukaryotic cells. Okay, so you need to be able to identify and describe the characteristics of a prokaryote or prokaryotic cell. Same words, but with eukaryote or eukaryotic cell in there. Okay, and be able to distinguish between the two cell types. So if I showed you a picture of one, you'd be able to tell me eukaryote, prokaryote. Okay, so bacteria are prokaryotes. No. Okay, so they're single-celled organisms. They don't have a defined nucleus. They have ribosomes and maybe small membrane-type organelles, but not truly membrane-enclosed organelles. Okay, and many possess cell walls. They generally come in 3 shapes. Okay, they come as spheres, known as coccus bacteria. Okay, they come as rods, known as bacillus bacteria, and they come as spirals, known as spirillus bacteria. Okay, so there's many different kinds of each shape. If you've ever had strep throat, okay, how many people have had strep throat before? Okay, strep throat is caused by Streptococcus pneumoniae, which is a type of coccus bacteria. So little spherical bacteria that infect your tonsils and throat and whatever and make it swell up really bad and feels like you're swallowing a razor blade every time you swallow. Yeah, okay, they're pretty nasty. All right, bacillus bacteria, this is what you get when you eat at Arby's. I've only eaten at Arby's twice. I have gotten salmonella both times. So I just assume that you always get it when you go there because that's what's true for me. Okay, so yes, salmonella. Okay, if you don't cook your chicken enough, that's what you'll get. Okay, salmonella typically resides on birds and reptiles in their flesh quite a bit. So if you don't cook it enough, you can get salmonella. Okay, and that is very, very nasty. Okay, like you feel so sick that when you go into the bathroom, you don't know whether you should sit on the toilet or face it. Yeah, pretty nasty. Okay, all right. And then there are like the spirals. Spirals are quite a bit more rare than the other two shapes. Okay, syphilis is an example of a bacteria that is a spiral. Okay, that is a sexually transmitted disease that makes you go crazy later. It's apparently what Napoleon had. Maybe that's why they lost the Battle of Waterloo. Anyway, okay, weird history kind of thing there. All right, but there are the different kinds of bacteria, but they have a fair number of things in common, mostly that they have the cell walls and that they have the no organelles or anything inside. Okay, so there's diversity in the shape, like we said. There's the cocci, coccus, okay, Streptococcus mutans, okay, Streptococcus pneumoniae, Streptococcus mutans. Mutans is the flesh-eating bacteria. Okay, you don't want to get that. Okay, in fact, we had a student here a number of years ago who's now a gold medal Paralympic athlete who had that, lost his fingers and feet and everything to it. Okay, but now is a Paralympic rugby player. Okay. Cole Medell, if you've met him, if you know who he is. Okay, he went to school here. We've got the rod shaped like tetanus. So if when you get your tetanus injections, okay, your booster shots, you probably got those in grade 9, right? Okay, that's a vaccination against that particular bacterium. Okay, and if you get that, what it does is it releases a toxin. In fact, that's how all bacteria make you sick. Okay, their waste products are toxic to us, and so they make us feel awful when we have them. The one in the tetanus produces breaks down special proteins in your body that allow your muscles to contract and relax properly, mostly relax, and so eventually you can't relax your muscles. That's why it's called lockjaw. If you've ever heard of tetanus called that, okay, it's because people would literally break their teeth by the time they were near the end or close to death because they could not unclench their jaw. Okay, you essentially almost suffocate because you can't ventilate your lungs because the muscles that allow you to breathe have to relax and contract in order for you to ventilate. Okay, so yeah, not a good way to go. If you step on a rusty nail, go get a tetanus booster. Okay, because that seems like a rotten way to go to me. All right, and then, like we said, the coil-shaped bacteria. Okay, so Treponema pallidum, which causes syphilis. Okay, there's lots of different kinds of each of those, and just those are the ones kind of off the top of my head. All right, now here's the thing: these are all very, very small. These are electron microscope pictures of these things. Okay, a bacterium might be 5 to 10 micrometers in size. Okay, so quite small. All right, bacteria are sensitive to salt, okay, and temperature. So we can boil unclean water, okay, and that'll kill them off. So if if you've ever gone hiking, usually you either treat your water chemically or you filter it with a very small kind of micron filter, or boiling it will always work, okay, and it'll kill those off, at least the ones that would make you sick. There are bacteria that can live in like hot springs and near hydrothermal vents where the temperatures are incredibly high. They can tolerate that. Those organisms, of course, would not survive in your body because it would be way too cold for them, right? But they can survive in some really hostile environments. The ones that would be the biggest concern to us would be the ones that like it about 37 degrees. Okay, so we boil unclean water, cook meat thoroughly, okay, to kill any bacteria. Also, heavily salted meats like jerky can remain unspoiled by bacteria for a long time. Okay, how many people have ever made jerky before? right? So you put in the curing salt, and then you season it to make it flavorful, okay? And then you dehydrate it, okay? And all of that helps to prolong how long it will last before it becomes spoiled by fungus, bacteria, etc., because they don't like those kind of conditions, all right? If you got a lot of salt and very little water, osmosis is going to be working against you, even if you're a bacteria. So it works pretty well that way, okay? Now, give you an idea here. This is, on the right, is a picture of a Petri dish that has had a swab done on it. So you can kind of see these little dots here are bacterial colonies. Okay? Each one of them represents where a single bacterial cell was placed on the agar. That's the stuff that's inside of it, okay, that they grow on. Each one of those represents where one of those was. Over the course of 48 hours, sometimes 24 hours, okay, that bacteria has divided over and over and over and over again until it made a colony of bacteria that was big enough to be visible, okay, to the naked eye. So if you've ever had a throat swab, okay, where you've gone into the doctors, you had a sore throat, and they stick the Q-tip down your throat and make you gag, okay, and then they do this. They smear it onto the Petri dish. Okay, the stuff that's in the Petri dish is called agar. It's a nutrient medium. It's like Jell-O in texture, not taste. Okay, it does have sugars in it. It has amino acids in it. All the stuff that a bacteria could love. It's like a bacteria smorgasbord. Okay, but it's there so that they can grow on it. They throw this in an incubator, and a day later they look at it and they go, "Yep, you got strep throat. You can have your antibiotics." Okay? If it comes up with nothing on it, they tell you got a viral infection. Go home and get better, right? Because we don't prescribe antibiotics for viruses, okay? Because they don't work on viruses. They only work on these things, okay? Living things. All right. This is an electron microscope picture of the surface of a recently cleaned cutting board. Yeah, it doesn't look clean. Okay? If you just wash a cutting board with soap and water, you're going to miss stuff. Because when you cut stuff on a cutting board, you cut the cutting board. And then pieces of whatever you cut end up inside the cuts. And you don't always get those clean when you wash it with soap and water and a rag. Which is why you should do what with your cutting boards? Well, I wouldn't say burn them, but you should bleach them, okay? Or you should have a cutting board for fruits and vegetables and a cutting board for meats, okay? And that's what I have in my house. I have a wooden cutting board that I use for vegetables and fruit and things like that because I don't worry about having to bleach it as much. They don't have as many bacteria on them, okay? I can clean that one pretty well, but I have a plastic one that I can cut chicken on, okay? Because I want to bleach that thing every time I use it. Because when you cut into that chicken, you cut into the cutting board and you jam a whole bunch of salmonella into that little slice you made, okay? And if you don't bleach that thing and leave it sit in the bleach for a while, okay, you run the risk of the next time you cut something on there contaminating it. Now, a lot of people say, well, if you're only using it for meat, you're going to cook the meat after anyway. Yeah. You're probably right, but I'm a germaphobe and I want to make sure, okay? But there are some people who have one cutting board. Now, when you're making stir fry, what do you cut first? The vegetables. If you only have one cutting board, you cut the vegetables first, okay? Then cut the meat. Because if you cut the meat first, you might as well just throw the vegetables in a bag of raw chicken. Okay, because you're going to get sick, right? Just little things, when you start making your own meals, these are things to think about, okay? If you don't want to be sick, okay, you want to cut your vegetables first on the clean cutting board, okay, and then use the cutting board for meat and then bleach it, okay, just to be safe, all right? But you can see on here, there's bacteria. This is fungus here, this red thing with the with this growing off of it. These are the spores, okay, of a fungus, right, things like that, yeah. So stuff grows in there, and it's microscopic, and you can't see it, but it's there. Are you turning into a germaphobe yet? They're everywhere. They're crawling on me right now. Okay, yeah. All right, bacterial structure. So bacteria have cell walls. They have different types of cell walls. That makes some of them easy to kill and some of them not so easy to kill. Okay, most bacteria are what we call gram-positive. Okay, they're gram-positive because if you do a gram stain, which is basically just you put them on a slide with a certain chemical, and they'll stain purple. Okay, if they stain purple, it means that their cell walls are made of something called peptidoglycan, which is a long protein type protein/sugar, okay, that their cell walls are made out of. Makes them pretty resistant to most things. Your body can generally kill them off, but it struggles with it, okay. These are killed with normal antibiotics, okay. So if you took penicillin, penicillin would get rid of these, because penicillin breaks down peptidoglycan. And then it's essentially like if you had a knight wearing armor and you took off his armor and sent him into battle. He's probably going to die. Okay, that's what happens with this. The peptidoglycan gets essentially dissolved or reacted with and disappears, and then it's just a cell membrane, and your T cells have no problem killing off those bacteria. Okay, so when you take penicillin, it's not killing the bacteria, it's making them easier for your body to kill them, right? And as a result, you get better. Other kinds have like a triple layer of cell wall. They've got a membrane, and then they've got a layer of peptidoglycan, and then another membrane around it, okay? So they have like a double membrane. You can see here in cross section, here's a membrane, here's the peptidoglycan cell wall, and then here's another membrane, okay? Those are penicillin resistant. You don't want those, okay? You have to take much stronger antibiotics that are much harder on your body, okay, in order to kill those off, okay? And this is the problem with overprescribing antibiotics. The overprescription of antibiotics has led to the emergence of penicillin-resistant bacteria. Okay, think about it this way. This is natural selection that happens in the course of one day. You take penicillin, it kills off all the bacteria in your body except for this one that had a random mutation that made it look like this. It doesn't die. What does it do? It reproduces, and bacteria reproduce real fast. Okay, so in the course of like 2 days, you're sick again, right? And penicillin won't work. Right? That was the problem with overprescribing. Doctors in like the 70s and 80s like, penicillin, take it, it'll make you better. And it didn't matter. If you had a sore throat, take penicillin, you'll feel better. Even if it wasn't something penicillin would have helped with. Okay? They told you to take it anyway because it was the miracle drug. And then, of course, that led to a lot more things being resistant to it. So now doctors don't prescribe antibiotics nearly as much. Making sense? Okay. Now, you can actually cause a bacterium to mutate from this form to this form very easily. Okay? In university, second year, I took a microbiology course as well as a genetics course, and in both of them, we did a lab where we put E. coli bacteria under an ultraviolet light for 30 seconds. Okay? And then we put them on a petri dish that had penicillin on it as well as all the nutrients, which should have prevented the growth of any bacteria because E. coli are not resistant to penicillin. But we'd created X-Men mutated bacteria by putting them under ultraviolet light for 30 seconds. Okay, think about that. You get in a tanning bed, right? That's essentially what we did to them. Put them in a tanning bed, gave them a tan, they turned into this, okay? And then we had all these bacteria growing on the petri dish that had penicillin on it, okay? They were completely resistant to it. Weird, huh? Doesn't take much. Bacteria, because they're so simple, can mutate very easily. Remember, they don't have a nucleus. Their DNA just kind of floats freely inside the cytoplasm where it can be damaged and altered and whatever else. So mutations happen very easily. Okay, so one of the reasons that soil bacteria are so important is plants can't get atmospheric nitrogen directly. So we've talked about bacteria that make you sick, but there's lots of good bacteria out there too. All right, not that you should eat dirt, because that's not going to be good. Okay, but there are important good bacteria in dirt, and we talked about this yesterday, that can take atmospheric nitrogen and turn it into nitrates that plants can use, like the beans. Okay, they had those little nodules on their roots. Okay, these are the kind of bacteria we were talking about when we talked about that. Okay. So those are good ones, okay? And you see those in like compost, okay, and in soil and things like that. So these are little sow bugs. These are not bacteria, okay? They're little insects. But in that compost, there would be lots of nitrogen-fixing or ammonifying bacteria that can change nitrogen from one form to another. And we're going to skip all that stuff about nitrogen fixation because we don't need to know about that. Okay. What do you suppose that's a picture of? Looks like an anthill. Okay. What do you think the little yellow things are? They are tetanus bacteria, in fact. It is not a rusty nail, but it's a good guess. That is the sharp end of a pin under an electron microscope. Okay, yeah, it looks sharp to us because our eyes can only resolve to a certain detail, right? Okay, the resolution we're capable of seeing doesn't see, it sees that it's pointed because it's very, very small, but under an electron microscope, it's pretty blunt at the end. Okay, and it's covered in bacteria. All right, so if you're ever taking out a sliver with a pin, heat it up first. Okay, take a match and run the match under it for a few seconds. Not enough to get it red hot, you know, and burn yourself taking this. Okay, you just need to run the match under it or a lighter under it, okay, for just a couple of seconds to kill those things off before you jam it into your skin and start digging out that sliver. All right, because if you do that without cleaning it, okay, or run some alcohol over if you got an alcohol wipe or something like that, okay, but you got to kill those bacteria off. Otherwise, you could make yourself, you could get an infection, okay, in the spot where the sliver was. Now, maybe you got it from the sliver, maybe you got it from the dirty pin, I don't know, but I'm a germaphobe, so clean the pin. Okay. All right, so prokaryotes can live almost anywhere, okay? They're very, very hardy. So they can live in hot springs, they can live in places that are hot, cold, salty, acidic, basic, okay? Any places where eukaryotic cells could not live. Eukaryotic cells, while efficient, big, good surface area to volume ratio, all that kind of stuff, have a very narrow range in which they can operate. Okay? They're far more sensitive to environmental conditions because they're more complex. I mean, the more complex the machine, the more likely it is to break down. Agreed? Well, it's the same with cells. The more complex they are, okay, the narrower the range in which they can work. All right, and to give you an idea of how many bacteria there can be, there are more prokaryotes in your mouth right now than all the people who have ever lived. Not the people on Earth right now, all the people who have ever lived. Okay, so that's like, what, 12 or 14 billion, okay, something like that. It's a lot. Yeah, pretty disgusting. Think about that the next time you're going to kiss somebody. I have never used an electron microscope. They see me coming and go, he'll break that. Yeah, no, I've never used it. They're more common now and they're much smaller than they used to be when I was in university, which is, I'm not going to say how long ago. They were much bigger and yeah. I've seen one. Yes, they gave us a tour and we saw one. Yeah. It was like computers back then. They were also a lot bigger, slower, and whatever. Yeah. All right, so this is generally what a prokaryote looks like. Okay, we got the DNA, the nucleoid region. It's just kind of a snarl. Okay, we got the ribosomes that are in there, and then you've got the cell wall. You've got pili. Pili are like little arms and legs, okay, that they can attach to each other with for conjugation. That's bacteria sex. Okay, they conjugate like this and they can actually exchange pieces of their DNA through the cell membrane. And then each one of them is different than they were before. And then when they divide in half, they've produced genetically unique organisms. Okay, so yay. All right, and then they have the flagella, many of them that allow them to move around. All right, the big thing is the way they make us sick is their waste products are toxic to us, and those waste products may affect our body in different places. Salmonella affect your digestive tract, tetanus affects your muscles, Streptococcus mutans affects your throat and lymph nodes, okay, things like that. Okay, prokaryotic cells only have one chromosome. We have 46. It's just a ring, okay, not that that's all that important, but they can go dormant for very long periods of time. Okay, and that's believed to have been what caused the curse of Tutankhamun. Okay, King Tut, when they opened up his tomb, everybody who went into the tomb died days later. Okay, and they're like, well, see, we told you not to go in there. We told you he was cursed, and you went in anyway. Okay, well, actually, now kind of more modern evidence would say that there was bacteria that had gone dormant within the tomb, possibly what King Tut had died of, who knows, okay? And when they opened up the sarcophagus, all the dust came out, and they inhaled these dormant bacteria, who then got into a place where, hey, it's 37 degrees, there's lots of water here, let's go, let's party, okay? And they made these people sick enough to die. Okay? So bacteria can go dormant for long periods of time, and then when conditions are right, just perk right back up again. Okay. Prokaryotes most of them are chemoheterotrophs with the exception of blue-green algae. Blue-green algae are photoautotrophs. All right, that's all you need to know about that. Actually, there's other slides, but that's it. Okay. Have a good weekend. We'll see you on Monday.